Well, welcome back. Did you guys have a good Tuesday? Excellent, excellent. Today, you have a double header from Dave. We're going to start with a lot of lecture this morning from 10 to noon. We're then going to take an hour for lunch and then come back to do some hands-on data analysis with real files. So I hope that this will be useful for you to have some theory about how proteomics works and some hands-on practice for how it plays out. We've got a sign-up sheet that's passing around, so feel free to add your name to the roster. This is actually the first topic on which I ever gave a college lecture many, many years ago when I was a postdoc. And over time, I've found that these slides have mutated every year with some sort of new information and so on. With today's lecture, I've tried to add in some new content that will, I think, give us better context for understanding this, because you're not bioinformatics students per se, you're doing molecular medicine, and that really requires that you have a good grounding in both the analytical chemistry of LC-MS, but also the bioinformatics of how we make sense of the data. So today we're going to cover both identification and quantitation, but we're going to do it pretty superficially. There are going to be lots of things that we don't talk about, like hunting for post-translational modifications or use of labeled standards, for example. Those things will fall out. We're going to talk about 4 different main topics in the before lunch session. It's going to divide into 2. We're going to have a 10-minute break in the middle, because I know having 10 minutes to let my vocal cords recover is also useful, and there are other things you can relieve in that time. So let's then talk about how this is going to lay out. We're going to start with analytical chemistry. What are the methods we use to get samples ready to shoot in the mass spec? And how do we carry out mass spectrometry, tandem mass spectrometry on these samples? We're going to talk about database search algorithms. These are probably the most essential part of bioinformatics for determining what peptides are present in a mixture based on MS data. After the break, we'll have filtering peptide-spectrum matches and inferring proteins. This is a subject that I think doesn't get as much attention as it should, but at some point you have to be able to say which of these hypotheses about which peptides are present do I actually trust. And for that we must filter the PSMs, and from that decide which proteins we're going to claim are present. And then finally we'll talk about quantifying. All right, so I start with this cartoon because it's useful to sort of put us in place, I think. We start with a protein mixture. What protein mixture are we starting with? Might it be, if we're dealing with blood, if we're doing biomarkers in blood, we might be working with plasma, we might be working with serum. They are very problematic places to start proteomics though, so I wouldn't advise that for a first experiment. Cell lysates are much friendlier for us to work with. So how many proteins might you expect in a cell lysate? Probably you would start by asking me what kind of species, right? If you're working with plants or trees, you're going to have a different expectation about that than if you're working with E. coli guts or yeast guts or human proteins. So if you have a bit of a tumor and you release the proteins from that, you're going to have a much wider catalog of proteins to draw from. So we have wildly complex protein mixtures. And it's not just the number of proteins that's going to challenge us, it's also their dynamic range, because some proteins are present in screamingly higher numbers than those that are present at low levels. One of the very first things we end up doing is to produce a peptide mixture from that. And the peptide mixtures are going to be more complex again than the protein mixtures that we started with. So why would we do that? And the reason is that we deal with peptides in tandem mass spectrometry because they're easier to separate prior to the mass spec, and they're easier to analyze within the mass spec. So generally speaking, when people talk about proteomics, they're not talking about the investigation of intact proteins, but rather peptides produced from them. Very frequently you'll see that people, especially those dealing with very, very complex mixtures, are going to make use of fractionation. Oh, here we go. So fractionation is useful for subdividing one big, very complex mixture of all peptides from a sample into those that meet different biochemical criteria. So you might, for example, push the pH higher, do an LC separation, and then treat each of those fractions as something to which, as an individual sample to which LC-MS will be employed. The trouble with this is that the more fractions you split each sample into, the more instrument time is required to make sense of that. And instrument time is always at a premium. Tandem mass specs are frequently in the neighborhood of $1 million once you throw in all the pumps and column equipment that comes with them. So naturally, the first priority for any lab operating a mass spec is to keep it busy. So there's often far more samples that want instrument time than the instrument can handle at any given moment. So one of the most essential questions you should ask when you're submitting samples to a proteomics core is about how soon is it likely that this can actually go on the instrument. So peptide fractionation then is useful for separating wildly complex samples to those that are a lot simpler that can be subjected to LC-MS. We have liquid chromatography, which is pretty standard really across this field. You'll find a huge number of people using C18 beads in skinny columns at a very low flow rate to produce a shallow gradient where the hydrophobicity rises, knocking the peptides off into the mass spec. Now, mass spec is not going to allow us to interrogate just everything. It requires that something be ionized for us to be able to analyze it in the mass spec. So for us, the most typical way to do that is electrospray ionization. Has anyone heard of MALDI as well? Okay, so MALDI is an alternative strategy, but it's not so useful in conjunction with LC. So generally when you have nano LC or micro LC, you're going to have electrospray paired with it. If your sample is dried down in a spot, you use MALDI instead. Okay, so those are both ways to get biological molecules turned into ions. At that point, everything else can take place inside the tandem mass spec. Typically, we'll have some sort of high-resolution mass spectrometry that will tell us which ions are currently available over the course of an LC run. So let's talk about time frame here. For a given mixture of peptides, you're probably going to have maybe a 90-minute or a 2-hour LC-MS experiment, okay? During that time, we're going to collect tens of thousands of spectra. So what's happening down here at the bottom is going to be happening again and again and again, multiple times a second, all right? So this first high-resolution mass spectrum is going to tell us what mass-to-charge values appear as dominant ions at this moment in time of the LC. Then we can choose a particular mass-to-charge value that has a nice intense peak, and we can isolate many ions of that peptide at that mass-to-charge value. We can then use collisional-induced dissociation, or CID, to bounce these peptide ions off of noble gases. This causes them to gain temperature, to gain energy, and they dissipate this by breaking peptide bonds. That produces fragment ions. So these fragment ions relate to whatever we isolated back here. So at this stage, we're just talking about kind of the old school tandem mass spectrometry experiment. We have a bunch of copies of some peptide ion. They break independently, and their fragments are then collected or measured in a tandem MS or a MS-MS. The terms mean the same thing. All right. So that's the big picture of the collection, but I think it's useful for us to talk about this in a few more stages. So let's start with what kind of biochemistry gives us a sample that we can work with. Almost all of this is going to be about proteins until we get to the very last stage. So we have to do something to extract proteins from a biological mixture. If I have a bunch of cells that I've cultured, how much of their total weight is protein rather than something else? All right, what else would we find in these samples that might complicate our analysis of it? Proteins are not the only things in cells, right? Please.
Lipids.
Lipids, great, sure. So the cells are practically, their borders are defined by these lipid bilayers. So yes, certainly there's plenty of lipid to be found in a cell. We don't want that going into the mass spec with the proteins. So yeah, we need to get rid of the lipids.
Salts?
Salts, yes, salts can be very adventuresome. You don't necessarily want to spray those into your mass spec. Because what you want is every peptide to be protonated, not bearing a sodium ion. So yes, salts are a problem.
DNA and RNA?
Bingo, yes. That sticky mess that looks like snot, you do not want to shoot that into your mass spec alongside the proteins. So we have some work to do to get the proteins by themselves. So I'm kind of arcing over a huge body of work here that I hope will be something that if you really care about, you can read about further in this paper down at the bottom. But we start with lysis. We want to blow up the cells, because if the cells are intact, we can't really get at the soluble proteins inside them. Centrifugation is useful for removing what I've called cell debris here. If you were involved in a sequencing experiment, you would call that sample. But for proteomics, you don't want all the nucleic acids and stuff around. Hopefully those are all gobbled up in the mess of lipid bilayer that gets spun out. Then we get to the stuff that we think about a lot more in the world of proteomics. We start with denaturation. Proteins that are all folded up are hard for us to analyze well. We would like to be able to chop these things up at some point. So we need to denature proteins. The most typical way that we do that is put them in a urea solution. So the urea causes the proteins to unfold. Disulfides are a problem as well. If we have a pair of cysteines that are holding hands, that's a bit of a safety pin holding the structure together, so we want to break those. Most typically we use something like DTT for reduction. MMTS is another example of this, but generally speaking, reduction is something that you see people using DTT for this purpose. And of course, once you've broken the disulfides, you don't want them reforming, because that can happen spontaneously. So we wanted to use alkylation to block off those sulfhydryl side chains so that they don't reform. Typically we use something like iodoacetamide for that. There are alternatives for almost all of these steps. I'm talking about like the mainstream of how most everybody processes samples. Finally, we get to the digestion step. And this is where in 90% of the cases you'll see that trypsin is used in order to digest the peptide bonds that occur after arginine and lysine. Now I've already said that there are some exceptions to every rule here. If you have two arginines or two lysines, or a lysine and an arginine that are back-to-back in the sequence, trypsin's not going to be very fond of cutting there. If you have a proline next to the lysine or arginine, trypsin's not going to be very fond of cutting there. If you have acidic residues nearby. So I guess what I would ask you to think about is this digestion process is somewhat probabilistic. By this I mean there are some peptide bonds that are very uncomplicated. They have nothing nearby to prevent trypsin from doing its business, and these parts of the protein are well exposed by the denaturing process. Those are going to get cut with high probability. Something that's in a bit of structure that's rather hard to unfold could be a bigger problem. So hopefully the urea and reduction steps have done all that they can to free up all these different regions of the protein. Even with all that said, there are simply some proteins that are just resistant to overall proteolysis. So it might be that a protein that produces great signal for mass spec is not necessarily abundant. It might just be really friendly to all of these steps that it's going to go through. And it ionizes well, because remember, if it doesn't take on protons, it doesn't make it into the mass spec. Okay, now we have the time dimension in the LC-MS-MS experiment. So what are we doing with LC? So we start with this concept that reverse phase liquid chromatography is used, again, in 90% of the cases that you see out there. So the concept here is that we have beads that are covered with long carbon chains, usually like C18. So these long chains are sufficient to stick most peptides to them. Some peptides are just so polar that they don't stick very well to this, but they're kind of a minority. We expect that most peptides from these digests are going to stick reasonably well to the beads. But they don't all stick with the same affinity. Proteins that have lots of lots of leucines and isoleucines and valines, or very large hydrophobics, are very likely to stick to these beads very, very strongly. Others, we'll stick to them at first, but as soon as you hit them with any amount of hydrophobicity, they're going to be competed off. So what we're creating is a separation in time based on the hydrophobicity of the peptides. The least hydrophobic peptides come off the column first. The most hydrophobic ones come off last. I mentioned beads. These are not large things because most of the time in most research labs we're talking about nanoflow LC. And that means that we have a column that's maybe 100 microns across, not some massive column like a millimeter, nothing of that size. So if you're dealing with a core facility that cranks lots and lots of samples through and really needs 90% uptime, for them they'll probably be using something like microflow instead. And the flow rate through those columns is higher, and often the column diameters are larger as well. But I said we were going to increase the amount of hydrophobicity flowing across these columns with time. Well, the way that we do that is that we typically have two different solvents, A and B, and we change the relative ratios of these two solvents over the space of an hour-long experiment. We have our equilibration step, we finally load peptides onto the beads, we do some conditioning at stage C here, and finally we start collecting MS scans as we gradually increase the amount of the B solvent, which is typically got a fair bit of acetonitrile in it. That's what causes the hydrophobicity. So over this space, we're collecting MS scans, and the instrument is fast enough that it can generate, say, 10 MS every second. Okay, so that's typical for like the Orbitrap class of instruments. That's how you get these thousands and thousands of spectra, of tandem mass spectra, that result over the course of a 2-hour experiment. Any questions before we move ahead? I realize that's a lot of detail. I just rocketed through a whole bunch very quickly. Short version, you got to get proteins out of your sample. You got to disrupt those proteins so you can digest them. You digest them with trypsin, now you've got peptides. They need to become ions and be separated in time. We're finally to the part where we talk about the mass spec. Okay, for this, I would ask you to remember, I'm not writing quiz questions, I think, but this is a quiz question I write all the time. How do the ions observed in a mass spectrum differ from the ions you see in a tandem mass spectrum? I'm showing examples of both here. At the top, we have a mass spectrum. I've talked a little bit about M over Z if I was speaking American, or M over Z if I were anywhere else. The M over Z is the axis on which mass analyzers separate ions. So this mass to charge value that we see here is our way of separating the ions that are present at this particular moment in time. What peptide ions are eluting from the column at this moment? That's what the MS provides us. So we have a mass to charge axis and we have an intensity axis. So you might have already subtracted out charge in your mind to say, well, we can now just determine the masses of all the peptides that are here. But the mass to charge is what we're actually measuring. So we should talk about this a little bit. The ions that we produce from electrospray are typically +2 or +3 charged. If we're dealing with moldy, it's more likely we get singly charges. But in this case, I'm talking about the LC electrospray route, and for that, +2 and +3 ions predominate. So if you have an ion that contains, let's say, 10 amino acids, 10 amino acids times the average mass of an amino acid, let's call that 110. So 110 times 10 is 1,110 mass. So if you've got just a 10 amino acids, you're dealing with something that's already more than 1,000 Daltons. If it takes on a double charge, then that 1,100 gets split in half. The mass to charge value is half of the mass in that case. If you have a triply charged ion of that mass, you divide that by 3 instead of by 2, and that controls where on this axis it will appear. But we often speak of these ions as though we have such a clear notion of what their charges are. How could that be? And one of the answers here is that typically we're using high resolution mass spectrometry for the MS scan. And in a case like that, the instrument has sufficient resolution that we can tell the isotopes apart. Even a single neutron is easily perceived as separate from the monoisotope when we look at the mass spectrum from an Orbitrap. It's just not a problem. Plus 2 and plus 3 is never going to challenge a high resolution mass spec from being able to discern the isotopes from each other. So if the isotopes are 0.5 apart, you know the thing is a plus 2. If the isotopes are 0.33 apart, 0.333, I mean, you'd have a lot of decimals with the mass accuracy we have today, then you know it's a plus 3. Is it possible you have a plus 4 or plus 5? Yes, yes. And most software today can identify those too. But the bulk of what we produce are plus 2 and plus 3 ions. And we can discern that from the mass spectrum. Yes?
Yeah, so this technique, the ionization technique that mostly causes a +1 charge, is it multi or?
Multi,
Yeah.
yes.
Is that better or?
Oh, that's a good question. I think what I would say is that there are plenty of situations that can only really be addressed through MALDI.
Okay.
There are plenty of situations that can really only be addressed through liquid chromatography, ESI.
Yeah, okay.
If you have a liquid flow of peptides, then it's almost always going to be electrospray we talk about. If you were trying to identify a single spot off of a gel, then MALDI is going to be completely ideal for the purpose.
Yeah.
Because you don't need to waste an hour and a half on a gel spot to know what proteins are there.
Okay.
So they represent very different uses of the instruments, essentially. Okay, good question. All right, so this is what the instrument saw, the mass spectrum, when it was trying to decide which tandem mass spectra would it generate. And it looked at these ions and said, ooh, there's a nice peak over there at 735, right here. Maybe I should sample that in an MS-MS. So the instrument will collect one MS scan and from it collect a bunch of tandem mass spectra representing, say, the top 8 or the top 20 ions that it hasn't seen MS-MS for recently. This is called data-dependent acquisition, and it was the standard for how everybody did this for a long time. It's now losing sway to the technique we'll talk about at the end of this. But it's a lot easier to explain, so I'm starting here. Okay, so the instrument decides which targets it's going to go after, which precursor ions it's going to go after. And these peaks don't represent fragments, they represent peptides. Peptides that were produced by trypsin digest. So the instrument will collect a bunch of ions, let's say at 735, tickle them with a little voltage to cause them to gain energy. They will break to form to form fragment ions. Those can then be measured in a tandem mass spectrum. So the tandem mass spectrum here, sorry, I started saying this a while back, and it's a little silly, but it is evocative at least. A tandem mass spectrum can be thought of as the death cry of a particular peptide. This peptide, in this case, had a mass to charge value of 898. We collected a bunch of ions of it, we shattered them by adding energy, and now these are fragments that resulted from the destruction of this particular precursor ion. So we have a precursor ion and fragment ions that resulted from it. I can't remember what the non-deprecated term is for fragments. We used to call them daughter ions. I don't know. Fragment ions is what most people in bioinformatics would call them. Yes, sir?
But if you use a precursor, in this case it's like 898, how can you have a mass that's higher than that?
Ah, very good. Okay. So this is the mass to charge of the precursor. How could any fragment from it be higher in mass to charge than the precursor? There's a difference between how you asked the question and how I answered it. Because what you said is, how could the fragments have more mass than the precursor? But they don't. They have higher mass to charge. This is a +2 peptide. It's broken to produce a bunch of +1 fragments. We could talk about why that happens, but it's a little beside the point, I think, for today. So, because the fragments have lower charge and lower mass, it is still possible for them to be higher in mass to charge, but not higher in mass. Okay, so these, in a tandem MS, in an MS-MS, The peaks that you see represent the fragments resulting from the breaking of some peptide. Okay. And we're not going to talk a whole lot about how fragmentation works because, frankly, people have written 30-page chapters about all the different ways that peptides break. It's a complex topic. But let's examine this 10 amino acid peptide, TSIIGTIGBK. This is a triplet peptide. You see it's got that nice lysine there at the end. So it's rational that you would see a trypsin cut after lysine. We have threonine, serine, isoleucine, isoleucine. We know the masses of all of these. Okay, so our serine, for example, has a residue mass of 87 Daltons. Isoleucine has a mass of 113. Glycine has 57. Threonine's 101. I should know this off the top of my head. I'm actually blanking on threonine, but that's fine. So when we break a peptide at a particular peptide bond, the ions that result, the fragment ions that result, are called B ions and Y ions. Do A ions and C ions and X ions and Z ions exist? Yes, but they're not produced in this type of fragmentation. So for now, I would just have you remember that B ions and Y ions are what result from collision-induced dissociation. We are calling the one at the left a B4 ion, and that's because it contains 4 amino acids. TSII together here represent the B4 ion. And the last 6 amino acids are what remain from that when you break this peptide ion. So the same action that causes the dissociation of this to produce a B4 ion is also producing a Y6 ion. It's called the Y6 because it contains 6 amino acids. In this case, we know that their masses have been measured as 415 and 572. We'll talk in a moment how we compute those masses, but the short version is that as you move from the B4 ion to the B5 ion, you're just moving one peptide bond to the right, you should see a difference in mass equal to whatever amino acid you gained. So the B4 ion should be 57 mass units from the B5 ion. Does everyone see why that's so? Because the mass of glycine is 57 mass units. So if we look 57 mass units higher, could anyone add 415 to 57 for me? Out pops the phone. 415 and 57. Just call it out when you get it. 472. All right. 472, that's a scratch for us. We actually didn't get a good fragment there. Well, let's look at the other side. Let's make a larger ion for the Y6 series. So 572, I'm now going to add the mass of isoleucine, which is 113. So 113 and 572. 685. Look at that. Brilliant hit. Okay, so one of the things you should notice here is that some of these fragment ions are going to be quite intense, but just because a fragment ion is possible does not mean you're always going to detect it. So if we were to say the Y6 ion and the Y7 ion are present, maybe we can add another 113 to this. 685 and 113 should give us 798, I think. 798. Okay, so the Y6, Y7, Y8 series is all visible in this MSMS. And that should give us some confirmation that this peptide sequence really does belong to this MSMS. Now, is it possible to read a sequence from an MSMS? The jury is kind of split on this one. And what I would say is that some of the great masters in the field of MSMS have created long, short courses properly, whole semester courses in some cases, on how to interpret the tandem mass spectra of peptides. It is a skill you can learn, and you can get pretty good at inferring sequence from MSMS scans. But it is a challenge to do so, and not all of the spectra are beautiful. This is an example of a good spectrum. Bad ones are much, much harder to read. Okay, But hopefully, what you picked up from that was that when you hypothesize a peptide is represented by an MSMS, you can then look up which fragments are detected for that sequence in that tandem mass spectrum, and from that gain confidence that this peptide really is reflected by this MSMS. Okay? So, database search is the big hit for bioinformatics, make proteomics possible. Database search algorithms are the standard method. They're not the only, but they are the standard method for how we match a peptide sequence to tandem mass spectra. So let's talk through all the different pieces that make that possible. We start with kind of the most important ingredient of our search space. Search space means we're going to have some universe of peptides we draw from that represent possible answers for any MSMS. And that search space is going to be defined by a set of rules that are how the search engine was configured, and also a set of sequences that we provided. So we're going to start with a protein sequence database in FASTA format. The most typical way people would do this is go to UniProt knowledge base, seek out the reference proteome for the species that your sample represents, and then they would download the FASTA sequence database directly from there. That works pretty well. In fact, a lot of search engines will download it for you, so they can make your life rather simple in that way. How many proteins might we expect for a given species? Well, it's wildly variable. If you're dealing with something like E. coli, there are only about 4,400 protein coding genes. If you download yeast, you're going to get about 6,200. If you download the human proteome, reference proteome from SwissProt, you're going to get quite a variable number of proteins depending on how you do it. It is typical in our field that people ignore the machine-approved sequences for the human proteome and just grab the SwissProt proteins for human. This is a really big mistake because that doesn't give you all of the different protein coding genes in humans. So what you should really do is download the reviewed and unreviewed reference proteome for humans, and that's got something like 80,000 proteins in it. Now we have how many protein coding genes for humans? Anyone? Yes?
20,000?
About 20,000. That's a pretty safe answer, yeah. Somewhere in the neighborhood of 20,000. And yet, because there are multiple splice variants possible for those genes, we actually have a big expansion in how many sequences are possible. And if you include not just reviewed and unreviewed sequences for human proteins, but also include SwissProt isoforms on top of that, you're above 100,000 proteins that are possible in humans. So that starts as our search base. Any protein in a human is one that we need to consider as a possible match for the MSMS scans we've got. On the bench, you use trypsin to digest those proteins to produce peptides. In silico, the software needs to do the same thing. It needs to apply a trypsin cutting rule in order to turn those input protein sequences into peptide sequences. So that's typically done by a trypsin digest rule that says any arginine or lysine is an acceptable cutting site unless proline is the next amino acid. That's the most common way that people define this. That doesn't take into account things like when you have a pair of arginines back to back, where the cut happens maybe isn't as random as all that. But we'll leave that aside. This gives us a list of peptide sequences. Now we don't need to compare every peptide to every spectrum. We'll get to that in just a moment. But I'm going to talk in the next slide about the PTM expander. If we look for post-translational modifications, we need to allow for the possibility that some amino acids may appear in modified form or in unmodified form. That can greatly expand the pool of peptides. This is an important part of our search space. Are we considering modifications or not? These decorated peptides are then possible mass ladders. I'll talk about that phrase a little bit in a moment. But we can project what spectra should look like if they represent these peptides. But the peptide mass filter is going to determine which spectra get compared to which sequence. I'll have a slide about that in a moment. And finally, I'm going to have a slide about how do we predict what the MS-MS should look like for a given candidate peptide to be compared to this spectrum. And finally, we have to have some way of saying, given the prediction of what the spectrum looked like and what we've actually observed for this spectrum, do they line up or not? So we'll have to talk about all of those things in follow-up steps. And then we're going to take a break because this gets exhausting fast. So let's talk about decorating sequences with PTMs. Phosphorylation is one of the most popular post-translational modifications to identify by proteomics. It's a very common type of chemical modification for us to look at. What do we know about where phosphorylation typically occurs in proteins, in humans, let's say? I've got some clues on the slide. Amino acids that have side chains of an OH group, serine, threonine, and tyrosine, are the most common places in human proteome for us to find these phosphorylation marks on proteins. So here you can see that some of these S's have been left as just S's, and some have had the asterisk added to them. This is kind of an old school nomenclature to say this serine with the star on it is modified by a phosphorylation, and this serine without one is not modified by it. Now, these are all the same peptide sequence. We see that, right? These are just a peptide drawn from alpha casein. Alpha casein is a very modified protein in cow milk. So what we see at the top is the sequence as it appears in the database. And in this case, we're considering as a possible peptide this sequence with no modifications made to it. The relationship between the three red sequences that follow it are that they all carry one phosphorylation. And that means that the serine, or threonine on which those appear has a mass higher than what you would expect for that serine or threonine normally. It gains 80 Daltons because that's how massive the phosphorylation of PTM is. So instead of thinking about 87 as the mass unit for that the first serine, we might consider 167, 80 mass units higher, for the serine when it is observed in the modified state. Okay, so the red sequences, as a result, all have a mass 80 mass units higher than the unmodified sequence. These three blue sequences bear two PTMs. They bear two phosphorylations on board. As a result, they have a mass 160 mass units higher than the base sequence. And then there's the possibility that all three of the PTM possible sites for this peptide bear the 80 mass shift, in which case the green peptide sequence at the bottom has 240 mass units more mass than what the sequence does at the top. Those don't all get compared to the same spectra, though. Because they have different masses, we don't have to consider all of those possibilities for the same MS-MS. What I'm trying to point out here is that some sequences, by allowing for more modifications, end up getting exposed to a wider variety of masses than those that don't carry modifications. The other thing is that there's some expense to going after these. If you include these in a search space, each of those modifiable sites is may be seen in an unmodified state or in a modified state. So there are two different states for each of these modifiable sites. There are three of them, and 2 to the third is 8. So there are 8 possible decorations of the sequence we must consider. So what we have is an exponential cost when we consider looking for post-translational modifications in database search. Okay, I talked a moment ago about data-dependent acquisition. This was the process where we acquired MS scans of peptide ions that were intense in the MS. Okay, so this is a depiction that comes from that. But importantly, the instrument remembers which ion in the MS was chosen, isolated and fragmented to produce each MS. It knows what it blew up to create this MSMS. As a result, we know that that peptide sequences, we know what mass they should have, and therefore we know which ones can be considered as a candidate for comparison to each MSMS. Typically, the data that we work with from our core facility have a mass accuracy of around 10 parts per million. And that is an extremely accurate knowledge of what the peptide's intact mass should be. As a result, we can ignore the vast bulk of peptides out there when trying to come up with an explanation for each MSMS. We only need to consider those that fall within 10 parts per million of the ion we blew apart to create this MSMS. Okay, that's the value of the peptide mass filter. We don't need to compare every possible sequence to every MSMS. We only need to compare the sequences to a given MSMS that agree very well on the mass that we blew up to create it. Okay. Now finally, we have these peptide sequences, and we need to model what the MSMS should look like if we blew apart this sequence to make them. All right, so let's imagine that we have a 6 amino acid peptide that's broken in the middle. If it started as a +2 peptide ion, when it breaks, the most likely scenario is that one proton goes with the N-terminal fragment and one proton goes with the C-terminal fragment. Those are shown by those green blobs up there. So if we're trying to determine the mass of the things, we need to remember that this is more than just 3 amino acids in space. There are 3 amino acids linked together, so the residue masses matter, and they're bearing a proton. We have to keep the proton in mind when we remember the mass of the thing. So, in the case of the B ion, we just sum together those residue masses, add the proton mass that's ionizing it, and then we know the mass of the thing. For the C-terminal or Y ion, we have 3 amino acids. We have to add their masses together. We have the proton riding on board, but there's something else going on here. You see there's an H added to the N-terminus of the Y ion and an OH added to the C-terminus. So when you're computing the mass of a Y ion, you need to keep in mind the amino acids that comprise it, and you need to add water, because H plus OH is water, and you need to add the protons that decorate it. Things get messy, however, when we take into consideration +3 peptides. If a +3 peptide breaks, what determines which side gets which protons? Is it likely that one of the fragments takes all 3 protons with it? Not so much. Protons don't like being in proximity with each other. And if you break off a fragment and you're trying to gauge the odds of all 3 protons going to one side, it's kind of a low probability event. So what we typically see is that when we break a +3 peptide, 2 protons go with one side and 1 proton goes with the other. You might say, oh, well then that's pretty easy. The larger fragment is going to get both protons. But in fact, it's quite a messy thing. We wrote a paper all these years back about trying to find determinants of when 2 protons go one way and 1 proton goes the other way. It's actually quite difficult to do. As a result, most search engines will consider for every breakpoint the possibility that both fragments appear in both +1 and +2 states, which means that for the same sequence at +3, you're going to have a much busier prediction of what the spectrum looks like than if that same peptide ion is appearing at +2. Okay, the result of this is a mass ladder, that we have fragment ions resulting from the breakage between the first and second amino acids, the second and third amino acids, the third and fourth amino acids, the fourth and fifth, and the fifth and sixth. This creates a ladder of fragments, and you can sort of walk the sequence from one terminus to the other based on B ions or Y ions. That's our prediction of what the spectrum should look like. There we go. Okay, and finally, we get to the scoring step. Having a prediction of what the MSMS should look like for a given peptide sequence, and an observed MSMS, we now need to say, are they similar or are they dissimilar? And frankly, given that there are more than 30 peer-reviewed publications describing search engines, the number of answers that have come about for how to score a spectrum against a prediction is a very complicated topic. So I'm going to give you a relatively simple answer based on match counting that uses what's called a contingency table. If you took biostatistics at some point, you probably remember contingency tables, but we'll try to talk about how those play out. So I have kind of 4 possible outcomes. I did predict a fragment ion at position 284, and there was a peak. That's a hit, right? So from a match counter perspective, every time we predict that a peak should be seen and we have intensity at that location, we scratch ourselves on the back and say that's a success, that's a hit, that's a match. It's also possible, however, that I predict that at position 284 there is a peak, but there isn't one there. That's a miss. So I predicted that there should be an intensity there, but there wasn't one. That's a fail. I think the counterintuitive part of this is that we need to think about the other possibility. It may be that based on the peptide sequence I'm hypothesizing to explain the spectrum, I think there shouldn't be a peak at 284. And it could be that I say there's no peak there and there's no peak there. That's also good news. You don't want the spectrum to have loads and loads of peaks at places you don't think should be occupied. Or it might be that you predict that there's no intensity at 284 and there is no peak there. Again, this is a success. So you can see that we end up with these 4 outcomes described here. Maybe I predicted a total of 30 peaks for this peptide sequence, which means I predicted a lot of places where there shouldn't be peaks as well. I can capture the information about all of that in basically these 4 counts. The times I predicted a peak was there and it was there, the times I predicted a peak was there and it wasn't, the times I predicted nothing was there and nothing was there, and the times I predicted nothing was there and something was there. Sorry, I realize that's all it sounds a little like double talk, but in fact, you need to think about all of those 4 possibilities to score these things well. Those 4 counts can then be plugged into the binomial or the hypergeometric distribution, and out pops a probability that says, how likely was this to occur by random chance? And I might want to think about how much probability was attributed to doing a match even better than this. I can add those values together and get a p-value. The probability that by random chance a match this good would have happened by random chance. Sorry, I'm repeating myself. One of the most common softwares used for identifying peptides uses a model very, very similar to this. It's called MaxQuant, and you'll certainly encounter it as you look across the literature. What comes out of this is the fuel of proteomics, the PSM. The Peptide-Spectrum Match is our explanation, essentially. We have this collection of MS scans. For each of them, the software has put forward its best guess for which sequence out of the possible set was the best explanation for each MS. Some of those are great, some of them are crap, but every spectrum has our best attempt to explain why this MS appeared. So, the Peptide-Spectrum Match is a hypothesis that this spectrum can be explained by this sequence. And that brings us to halftime. So I would suggest that we all gather back in here by 11:02. So take a moment, and soon we'll talk about how we figure out which of those PSMs are actually reliable. Okay. Okay, so for part 2, we've already talked about how the peptides are produced from biological samples, how the peptides result in spectra and tandem mass spectra, and now we've talked about how we create a hypothesis about what peptide each MS represents. But we are not done. There's a lot of work left to do, and frankly, a lot of the emphasis in the proteome informatics field has moved to the tasks that I'm talking about in this latter half. So, if you think of the first part as the essentials of how we identify stuff, there's still a lot of work left to do, because remember, people care, most of the biologists sending samples to the core facility are not actually interested in peptides at all. they're interested in proteins. So we need to be able to talk then about how we get to protein level information, confident protein and level identification information, and how we quantify them. This talk is going to necessarily be pretty superficial because there are a lot of pieces to all of this that can complicate the story. But I hope that we can talk about what we do with those PSMs to find a confident set, how we make protein inferences that explain the existence of these peptides in our mixes, and how we can go about quantifying them. But let's start with those search scores. You remember that we were talking about a contingency table from a match counting score algorithm, but I want you to remember there are a bazillion ways that you can decide whether the spectrum you predict for a peptide matches one that you've observed in the MSMS. So we need to be a little generic here. We need to think about some of the folks doing this are using statistically derived methods. Some people are using methods derived from mechanical engineering. Cross-correlation is a very popular method for comparing an observed spectrum to one you've predicted. There are lots of ways you can go about doing it. Some of the computer science methods that have come up are very nearly machine learning-like in how they proceed to score the similarity between an observed and a predicted spectrum. But what role do we need to accomplish by using these search scores? So we're going to start with kind of a lesser understood concept here, but it's actually rather an important one. Generally speaking, if the database search engine doesn't get the correct sequence all the way at the top rank for the MSMS, we're probably going to ignore that MSMS. It's probably going to be treated as a failed identification. So being able to rank the one true sequence for this MSMS all the way at the top is almost a prerequisite for that peptide being identified correctly. It actually shocks people sometimes when they realize for the first time that at least 10 years ago, if you identified even 10% of the MSMS scans you acquired, you were doing pretty well. That's changed a lot, largely due to having high resolution measurements, not just of peptide ions, but of the fragments that come from them. And if you're dealing today with a microbe where you can grow up as much of it as you want and you don't have limitations on how much sample is available to you, and you have a pretty small proteome to work with because you're dealing with microbes rather than mammals, you can actually identify as many as 50% of the spectra that you collect on the instrument today. And that's rather good. You should not expect that if you're dealing with biofluids. Plasma, serum, the numbers are going to be much worse for you. But in the best case, you may be able to identify as many as 50% of the tandem mass spectra you acquire. So let's talk about how we get there. First off, search engine gets the correct sequence ranked all the way at the top for each MSMS. But I'm going to talk less about the glorious, gorgeous spectra now and talk about the reality. Pushing the correct sequence to the top for marginal spectra is the tricky bit. Almost any search engine you use can, if given a gorgeous tandem mass spectrum, give you a correct answer about what peptide it represents. But it's how you deal with the stuff in the gray zone, where you have a peptide that's barely standing out above the noise, the chemical noise of a mixture, that it gets really, really difficult to push the correct sequence to the top. You still rely on having a good fragment series, good series of B ions and Y ions in the MSMS, or identifying it is not going to be very successful. Dealing with marginal spectra then really separates the good algorithms from the poor ones. Okay, but this is looking at things from the fate of an individual MSMS. I would say that what we actually care about is our aggregate performance. We're not really focused on the information we get from a single MSMS. We want to know how we're doing in the great collection of all the MSMS scans. So for that, we need to be able to filter the peptide-spectrum matches to reflect just those we actually have confidence in. So we're going to use the search engine scores to separate the good stuff from the bad stuff. I'm going to mention, though, that we're going to be using more information than just that provided by the search engine. Well-calibrated scores are those that you can use to threshold across many spectra. If you have a score that depends very heavily on some physical phenomenon of the peptide, it may not be that useful. To use an example, in the old days, it was very common that people were using the sequest search engine for producing loads and loads of IDs. But sequest had a length dependence in it. If you had a very short peptide, the cross-correlation score that resulted from it was in a very different domain than if you had a peptide that was 20 amino acids long. So how do you deal with a length dependence in trying to use this score to separate the good stuff from the bad? So being able to calibrate scores so that they're able to be used to threshold across spectra on an even-Steven basis is really important and valuable. So for the moment, let's step away from any one search engine out there. I'm going to use Dave's theoretical search engine for just a moment. Here we've mocked up a bunch of scores in R using a simulation. So let's say that among this collection of peptide-spectrum matches, I have an equal number of correctly identified and incorrectly identified spectra. I have a mean value of 3 on, we'll call it 3 Daves. A score of 3 Daves is the mean for incorrectly identified spectra, and a mean of 6 Daves is the average score when we have a correctly identified spectrum. And I'm just going to throw a standard deviation of 1 on both of those. Now I said that there were 10,000 correctly identified spectra and 10,000 incorrectly identified spectra. I'm using a target decoy method to evaluate whether these scores are good or not. Now, this is very, very commonly used in proteomics. This is not just part of Dave's imagination. This is something used almost everywhere in database search. We double the size of our sequence databases every time we run a database search. So if you're doing a human database search, you have 100,000 sequences, and now you have another 100,000 sequences that have all been reversed N terminus for C terminus. So when we compare peptides to the spectra, half of the comparisons are to totally bogus non-biological sequences, because these reverse sequences do not exist in nature. The value of doing so is that every time we match to these reversed or decoy sequences, we know that that is a false match. We know so because it is a reversed entry and it's marked as such in the sequence database. So if we correctly identify a peptide, if we correctly identify an MSMS matched to a peptide sequence, we know that it must be to a target sequence. It must be to a real, biologically meaningful sequence. All of the correctly identified sequences will be to targets. And that's what this hump over on the right represents. The hump over on the right is mostly comprised of correctly identified sequences, and all of those have to come from the target database, the database we downloaded from Uniprot. However, if a spectrum is falsely identified, it's not guaranteed to come from the decoy database. It's just as possible to falsely identify a spectrum to a bogus peptide hit using target sequences as it is decoys. So in effect, we have kind of a coin flip. A falsely identified spectrum has just as good a chance of matching to a decoy sequence as it does to a target sequence. So the left hump, the low-scoring, falsely identified spectra, is comprised of both decoy and target hits. Now, we didn't say that everything correct scored a 6 and everything incorrect scored a 3. What we said is that the mean of correct scores was 6 and the mean of incorrect scores was 3, but they have the standard deviation around them, which means that there's this mix between these two peaks. We might start with the question of how could we protect ourselves from any false hits? Well, we know that the highest false hit scores somewhere around, I'm going to call that 6. So if you set your threshold at the highest score achieved by a decoy sequence, your estimate of how many false hits you've got is 0. So I now lay a line across here and I say, you must be higher than this line for us to accept you as a positive hit. That's great. Having no false matches is good. But something that should be really obvious to you right away is if you lay a line across this data set here, there's a lot of true stuff that you're throwing out. There are a lot of real peptide hits that you lose if you try to cut your false discovery rate to 0. So instead, we need to find some sort of balance between falsely identified and correctly identified spectra. The typical way that this proceeds in proteomics is that people set some upper bound on the aggregate error rate of their data collection. So you might say, I'm willing to accept that up to 5% of all of my PSMs that I report may be false. So now I need to find a place to set this line so that I'm able to achieve a mix where my estimate of how many falses there are is about 5%. More conservative approaches would say, I don't want the false discovery rate of PSMs to be worse than 1%. But you can see that that means I'm going to have to scoot the line back to the right a bit to achieve that. Okay, oh yes,
Yeah, I have a question still.
yes,
So I don't fully understand, so when you reverse the sequence,
yes.
and that sequence is overlapping, so it's the same with the normal non-reverse sequence, then it gets the decoy label.
The protein sequence
Yeah.
for a real protein
Yeah,
is given in reverse order.
yeah,
C terminus first, all the
Yeah.
way to the N terminus. And they exist as separate entries in our FASTA database. One is called, say, beta gal,
yeah.
and the other one is called beta gal underscore rev,
Mhm.
to indicate that it was the reverse of that
Yeah.
sequence. So anytime I match a spectrum to beta gal rev,
Yeah.
I know it's a falsely identified spectrum.
And why is it necessarily false when they match? That's maybe one question.
Okay. Any MS has some predilection for matching to a random sequence. Most of the tandem mass spectra we collect have, say, 200 peaks in them. And even the correct answer is not going to explain all 200 peaks. Hopefully it'll explain the big ones, but there will be plenty of background noise that will be left unexplained. So if I match an arbitrary sequence to it, there will be some amount of hit rate. And I'm going to compare tens of thousands of sequences to this spectrum. So the chance that an incorrect sequence will outcompete the correct sequence is real. That can happen. Or it can simply be that this spectrum doesn't have an explanation that's in the search space. What if I'm dealing with a human gut sample and I included human proteins but I left out microbes? I mean, maybe I've got some E. coli growing in my guts. Those peptides are real. They fragment, they produce fragment ions. But if I haven't included E. coli in my search database, I can't identify it correctly. So now this spectrum is going to get matched to something. Something will produce the highest score. It just may not be an impressive one. So having the ability to estimate the fraction of our PSMs that we've accepted that are bogus is really, really important. These reversed entries are going to give us the ability to spot them. Okay, so these are the data from my simulation in graphical form. Now I'm looking at them in Excel. First rule of bioinformatics, don't do it in Excel, but sometimes you just do. All right, so here I have sorted my list of PSMs from my simulation, and the ones that are blue come from the target databases, and the ones that are white are actually from the red distribution, they're decoy hits. And here I'm looking at hits 6,676 through 6,698, just out of this giant list. And what I want to show you is that at this position in the list, most of them are still target hits. Up at the top, they would all be blue, because there aren't any reverse hits at really, really high scores. But once I get down into the interesting part of the list, yeah, there are decoys that I'm hitting. As I get lower and lower in that list, the number of decoys gets denser and denser in that list. So when I make my estimate of my false discovery rate, I'm going to be dropping kind of a bar down this list, and I'm trying to find where is low enough for good sensitivity, but high enough for good specificity. Basically, I'm trying to find how to counterbalance good possibly real hits without doping too many false decoys in the mix too. Okay? The way that we do that typically is simply to double the number of decoys. Why do we do this? We do this because a falsely identified MSMS has, to some approximation at least, the same probability as coming from the decoy sequences as it does from a real one, from a target sequence. So a spectrum that is falsely identified has equally good shot of coming from targets as it does from decoys. So anytime we see a decoy, we assume that it's kind of standing in for another silent false match that's buried among the target sequences. So by doubling the number of decoys we have above our threshold, we're making our best estimate of how many of what we've kept so far are false hits. And then we can just ask how many total PSMs are above the list as our denominator. We can then double the number of decoys by the total number of PSMs over the threshold, and that gives us our false discovery rate, our estimate of what fraction of the accepted PSMs at this threshold are bogus. I'm not really going to get into the Q-value thing at the moment. But basically, if you have some target false discovery rate you want to hit, you're going to float this line as low as you can, but keeping that false discovery rate. And, as in all things, to achieve more sensitivity, to explain a larger number of spectra, is always going to come at the cost of a higher error rate. So, I'm going to talk about an uncomfortable reality for just a moment. In proteomics, the PI who is running the show always wants the longest protein list possible. There's not a PI alive who doesn't want to say, I identified 5,000 proteins rather than 2,000 proteins. So there's always pressure in proteomics to give the big number. Number go up is good. We love this. But the reality is that we have to be rather careful in how we control false discovery rates, because you don't want to send some PhD on a 5-year mission to chase a false positive. We can agree this is a bad outcome. We don't want to waste researcher time and resources on something that's a false match. So we have to be careful that we limit our error rates among our identifications to a trusted set of identifications. But we're not just going to use search scores for this purpose. And we're not going to use kind of a simple ordering of all of the spectra by search score as our final determination of what's real and what's not. There's a lot of information to be found that isn't coming from the search engine. So we start with the first one. How closely do we match the precursor or the peptide ion mass between what we claim it to be and what we observed? Are those numbers 1 ppm apart? Are they 5 ppm apart? Are they 20 ppm apart? Those are very different classes of error. So that's not captured in the search engine score, but it's certainly useful information when you're trying to separate a good hit from a bad. Getting a more accurate precursor mass calculated from your sequence is a good thing. What about trypsin specificity? If you said trypsin produced this peptide, and I've got a nice trypsin cleavage site on the N-terminus and a nice trypsin site on the C-terminus, that's pretty good. We like that outcome. But sometimes you can set up your search to consider the possibility of semi-tryptic peptides, those that are tryptic on one end but anything on the other end. When you look at a case like that, it's much more believable to accept PSMs that represent fully tryptic peptides, not those that have anything goes on one end. So trypsin specificity is the kind of thing we can use for deciding which PSMs are acceptable and which are not. If you have a PSM that has loads of post-translational modifications on board, and that's the only way you could identify it, that's not a good thing. If you do a phosphorylation search, and this peptide that your PI is so excited about bears 4 phosphorylations on it, it's not trustworthy, not really. Sometimes those are real, but frankly, it's quite often the case that the software uses post-translational modifications to force a fit between a sequence and an MSMS. And those aren't so trustworthy. Okay, what about some of these things that we can simulate about peptides? You'll see an example of this afternoon. At what retention time would we expect this peptide to elute? Now that's not in the search engine score, and really I haven't talked about retention time at all. But what if Sequest or another search engine tells me that I have a highly hydrophobic peptide, a long one that's just littered with leucines and valines and tryptophans and so on it, and this thing came off 12 minutes into a 2-hour run. That's garbage. It's garbage. A highly hydrophobic peptide like that is something you're going to have to pry off of the C18 column with a crowbar. You're going to have to hit it with a huge amount of acetonitrile. It's going to show up at maybe minute 100, but it's not going to show up at minute 12. So you can use retention time predictions to figure out whether a peptide is real or not. And you can even simulate with a high quality what this MSMS should look like if this peptide sequence were real. We're stepping well out of match counting here. We're not just saying, well, we went looking for 30 fragments and we matched 12, so I guess it's real. Instead, now we're able to say the most intense 5 fragments for this peptide should appear at these M/Z locations. Were those the most intense in the spectrum or not? That's going well beyond what a search engine is going to do to validate the ID. So this information is all quite valuable. Now I'm going to mention this last one because it's something that actually has tripped up the machine learning community quite badly a few years back. If I have a peptide that came from albumin, I should have a higher prior probability on that than if it's from some protein I've never heard of. Why do I say albumin? Because albumin shows up everywhere. Practically any human sample you generate, there's going to be loads of serum albumin in it. The protein is just not stoppable, and it's very friendly to mass specs. So to see a peptide from albumin and say, oh yeah, it's just albumin, that has a really high probability of correctness associated with it. Whereas some random protein that's expressed at a vanishingly low level and only ever gives you 2 peptides if you're looking for it, that protein has a much lower prior probability associated with it. But if you were to use how frequently was each accession seen across all of the PSMs, irrespective of their score, and use that to guide you in what you accept, suddenly you haven't, you've destroyed the independence of our judgment about this PSM. Now your willingness to accept this PSM is based on your decisions about other PSMs. And that can really, really lead to bad overfitting situations in machine learning. And machine learning very frequently shows up at this stage as we try to pull all of this information together. Okay, so that one we don't use. And now let's take a brief moment to think about something else. We started by digesting proteins to produce these peptides. But if the biologist actually cares about proteins, why aren't we studying the proteins directly? Can you separate proteins in a column? Yes? You don't want to use C18 for that. You'd use something like C4 material. But you can separate intact proteins, proteoforms, using liquid chromatography. Can you fragment ions of an intact protein in a tandem mass spectrometer? You can. Do they fragment well? Not always. But there are some specialized instruments out there that have really improved on our ability to make good fragments from these. Methods like electron transfer dissociation rather than collisional induced dissociation can give you much more informative spectra, for example. So, if what we actually care about is proteins, why aren't we just working with top-down proteomics? It's a good method. The sensitivity that we achieve in shotgun proteomics, in the bottom-up peptide-based proteomics, is much, much better than what you can achieve with top-down. But if your biological question is about the intact protein, about the correlation of different parts of that protein, then you should be thinking about this method instead. So what I want you to remember is that in the shotgun world, in the peptide-based mass spectrometry, the information about which proteins are found is inferential. We don't have direct evidence that this protein is present. We have evidence based on its peptides. And that is a rather crucial distinction. The peptides don't remember what protein they came from. The peptides are separated from each other over LC time, and unless you're looking at a particular gel plug that has all the protein kind of isolated by itself, if you're using LC, that information of the connectivity among the peptides is lost. So this is called a bipartite graph. It has objects of two types. It has proteins. These are hypotheses again about which proteins may be used to explain peptides, and the peptides are shown over here at the right. Now I've drawn an arrow from some of those boxes to some of those bubbles at the right. Those arrows say this protein could be used to explain the appearance of these peptides. This is kind of a nasty situation, actually. This is data from a xenograft. Has anyone heard of a xenograft? Okay. In the field of cancer, we very frequently want to do biology on tumors, but doing experimentation on people with tumors is considered rude, with good reason. So instead, we will often take a few human tumor cells and inject them into mice. And I mean, we can be gross to mice, and I guess no one cares very much. Sorry, I've always been squeamish about killing animals in science. I'm not a very good scientist in that respect. But so we have now a mouse with human tumors growing in it. But the tumor tissue we take from that mouse will have proteins from both humans and mice, because the stuff circulating through there is generated by the mouse, but those cells grew from human ones. So in this case, we now have this set of proteins that represent different isoforms of related genes, and they come from both human and mouse. How do we figure out which proteins are real from this set? There is not, I would say, a firm consensus in the field on how to do this. But the short version that most people have come to is that parsimony is valuable in not overstating what your evidence can tell you. Parsimony means we want to use the smallest set of proteins possible for us to explain all the peptide evidence we have. Parsimony. Have folks heard of Occam, William of Occam?
No.
Ah, okay. Great medieval thinker who said that if you have a simple explanation, he didn't say, he wrote in Latin, right? So this is, I'm telling a just so story here. He said, if you've got a simple answer for something, that's more compelling than if you have a complex, unnecessarily messy answer for something. You know, if you come home and learn that hear the neighbors say that your kids painted my cat, you know, the kids will tell you one story and your neighbor is telling you another one. The kids painted the cat might actually be the right one. You know, if they start talking about the aliens who showed up and painted the cat, well then their story has become unnecessarily complex. In the same way, if you can explain the peptides you've observed with 3,000 proteins or 5,000 proteins, the 3,000 answer is probably being the more parsimonious one, has a better chance of being accurate. So parsimony is one of the ways that we go about this. In this case, if you simply invoke these two proteins, the arrows coming out of them account for all of the peptides. So this database has a lot of possible relationships between protein sequences and peptide sequences, but just two of those proteins are enough to explain all of the peptides at the right. That's a parsimony approach for naming proteins. Okay. That's the end of the identification part. We're going to move into quantitation. This is necessarily quick, I'm sorry. But I hope that we can cover enough about quantitation that you have a clear notion of how we go about quantitating in proteomics. One of the little jokes mass spectrometrists tell on themselves is that we are masters of the x-axis. We can tell you the mass of things and the mass to charge of things very, very accurately. But as soon as it comes to those intensity values, things get a little hinky quickly. Those numbers can bounce around quite a lot. An intensity value nominally tells us about how many ions there are of a thing. But different chemicals at different mass to charge values have very different ionization potentials. As a result, the intensity of an ion may be larger than the intensity of another ion, and yet there may be less of it present. It's just ionizing better. So we have to be relatively careful then when we start talking about, start making bold statements like, if the intensity is higher, there's more of it there. If you're comparing for the same ion, that's true, generally. But then we would need to think about things like linearity. You know, if the signal that you receive for an ion doubles, does that mean there's twice as much of it there? If you're in the region of linearity, yes. If you're outside it, no. So let's talk then about how intensity values become quantities and what we're doing to rearrange mass spectrometry to make it easier to get stable intensities, thus stable quantities. Okay, the extracted ion chromatogram is our best friend in most of proteomics. The extracted ion chromatogram asks, what is the intensity observed for this ion as a function of retention time? There's kind of a rule of thumb that in most LC setups for proteomics, you have about 30 seconds to capture information about a particular peptide. So if you don't capture a tandem mass spectrum of it in that time, you don't see it. And if you aren't capturing lots of mass spectra, you may not have a very clean chromatogram seen for it. So the extracted ion chromatogram means that we're grabbing just the intensity values for this particular ion. We're not paying attention to all the other intensities seen in the spectrum, just focused on this particular target. Okay, so maybe you have 2 hours of LC time in which you have, say, 30 seconds for the peptide. We're able to trace its intensity from the MS scans as a function of time just during the interval it actually appears. We typically would try to integrate that signal to make an area. The area would give us some indication about quantity. But we don't compare quantities among peptides. We look at it for this peptide. Is it possible to quantify something if you don't identify it? Again, this was kind of a controversial concept, but people have decided you rather well can. It's quite possible that the ion produced a speed bump in the MS scans, that the intensity for that peptide appeared, rose to a peak and then fell off, and yet it was not intense enough that it was sampled for MSMS. The instrument is busy, right? There are dozens of peptides coming off this column at any instant, and so it might be that the instrument never got around to collecting an MSMS of this peptide during the 30 seconds it had to capture it. And yet, if you look at the MS scans, you see that mass to charge value and its intensity shows up at the same time as in other samples and then goes away at about the same time. So we need the ability to deal with minor variability in retention times, the time at which peptides appear from LC, because there's just some amount of variability in it, that run the same column on the same sample every time, you're going to see maybe as much as a minute shift in when a peptide shows up in its chromatographic time. So we have some sort of alignment routine to help us to adjust so that we're kind of looking at even-steven times among different runs. And we count on the fact that in at least one of these runs, we did have an identification for the peptide. You have to have some basis for saying that this XIC value represents this peptide. So we have to identify it somewhere. But we can then extend from the run where it was identified into a run where it wasn't by having aligned retention times. We can then use the information that in run 2 this was identified to a particular peptide to say, it wasn't identified here, but it's the same mass to charge and it's the same retention time in this other file. So we can use this to fill in what would otherwise be missing values. And if you haven't done a lot of biostats yet, let me just say missing values stress us out. If you say that this peptide was present at some low quantity, that's generally more useful to you than saying, I have no data for this peptide in this run. Okay, so it is plausible that you could try to quantify a peptide that wasn't identified in this run, find something that looked like an extracted ion chromatogram and write that down as its intensity, but it's not even the same peptide. That can happen, because remember, this is a crowded space. We've got tens of thousands of peptides all sharing the same M/Z space and retention time space. And so it's quite possible that you would go looking at a random location in this run and find something that looks like an XIC, but it has nothing to do with the peptide you thought it was. If that happens, you have a false quantitation event. And that could mean that even though a protein is categorically not present in the sample, you may still impute, you may still inappropriately quantify it even though it's not there. Worth knowing that that happens. Okay, now, so far I've been talking about extracted ion chromatograms as something that we see for peptide ions in the MS scans. But it is also possible to create chromatograms for the fragments observed for a peptide, so long as you measure them at multiple time points. So what you get is something that looks like this. This is some popular software called Skyline that's very good for this kind of thing. Here we're looking at a selected reaction monitoring experiment. It's an SRM style experiment. It's a kind of targeted quantitation. The instrument here has been set up to look for any evidence of the fragments from this particular peptide across all of this time. And here we see the Y7, Y4, Y6, Y3, and Y5 ions all rise together in time and fall together in time. The reason for this is that the peptide has a characteristic retention time. And during that 30-second interval, that's when the fragments for it become visible and go away again. So we can use these effectively as a proxy for the protein's intensity. The fragments for it are rising and falling together, all at a time of around 51.8 minutes in this case. I think it should be apparent that if you have chromatograms for multiple fragments for a peptide, you can quantify it more reliably than if you're relying on just the intact peptide ion chromatogram. We have more information to associate with its sequence by looking at the fragment ions. So if you can get chromatograms for fragments in addition to chromatograms for the intact peptide, that's a really good thing. And this brings us to data-independent acquisition, a very popular way to go after this. I should define this term though. Transition is something that shows up very frequently in this topic. A transition says, I had an ion that had a mass of X, and it fragmented, and it produced this ion with mass Y. The relationship of X and Y came from blowing apart X, is a transition. So when we look at this, we have 1, 2, 3, 4, 5 transitions all measured for the same peptide. It's a useful concept to kind of organize the relationship between something that you isolated and something that resulted from its breakage. Okay, now I don't always talk about DIA in identification and quantitation classes, but in this case we really need to. If you send a sample to the Interfaculty Mass Spectrometry Center, you're going to be getting a DIA experiment in all likelihood. Instead of producing data the way that I've been describing, the DDA, the data dependent acquisition method, what we typically see in use here is data independent acquisition. And that's worth talking about separately. This is a complete reorganization of the way the instrument is going to collect data. I've been talking about MS scans, about mass spectra several times over. Those are represented here by the orange bars. So we have an orange bar followed by a whole bunch of these blue bars and then another orange bar. So this is 2 cycles of what's called a DIA experiment. The way that a DIA experiment changes is that instead of looking to collect tandem mass spectra of the ions that are intense in mass spectrometry, we're going to do a collection of MS scans that range in large tile spaces, essentially. Here we have isolation windows, this is a key term with this, that are 24 mass units wide, which means that instead of isolating a whole bunch of ions of one particular M over Z value, we actually have this window that's 24 mass units wide. Any peptide that falls within those 24 mass units, we are going to co-fragment and measure the fragments all together. Well, that might seem a little bizarre. I've just gone through all this time talking about tandem mass spectra, representing the death cry of a peptide, but now I'm talking about mixing them all together. All of the peptide ions within a 24 mass range, a 24 Dalton mass range, or Thomson, are going to be co-fragmented, and their fragments will all coexist in a single MSMS. That's really busy. But this is really valuable because we can make this pass through the entire mass range with 25 MSMS scans in about 3 seconds, say, which means that we can come back and measure it again 3 seconds later, and then another 3 seconds later, and then another 3 seconds later. Now remember our rule of thumb, a peptide is visible for about 30 seconds. If you can measure it every 3 seconds, you might get 10 or 11 slices across its chromatogram and be able to measure both its precursor, the intact peptide in the orange MS scans, and its fragments in these blue scans. Amazingly, bioinformatics people have figured out even how to do identification directly of peptides, even when their fragments are all mixed together with other nearby precursors. So this has become a very, very common method, and we found that we're able to identify even more peptides from DIA-style spectra than was possible from DDA. That was something I didn't believe for a long time. There is, however, a big, what people call DIA from lab to lab can differ quite a lot. Some people will do lots and lots of these isolation windows, and some people will do relatively few. As a result, if you have wider windows, even wider than 24 mass to charge, then that means even more peptides are going to be sharing the reporting space of each MSMS. If you make them narrow, the time required to collect all of those windows as you shoot across will rise. As a result, you'll have a little less granularity in the measurements of intensity with time. Okay, so why would we do this? Why would we? We start with multiplexing. All the peptides within the defined ranges of M/Z are co-fragmented with frequent MSMS for each window. We're not really in a place where we have to treat each MSMS as an independent thing. Now the evidence, the fragment evidence for a given peptide is observed multiple times because those MSMS scans are frequent for a given isolation window. This is fixed sampling, and this is something that people have complained about for years about shotgun proteomics. The idea being that if the instrument is collecting MSMS for just the intense peptide ions, and the intensity observed for peptide ions changes a lot, well, then we may get a completely different selection of which MSMS scans are acquired for a given sample. I was part of a study that documented the extent to which ions were sampled for the same mixture shot 8 times on different instruments. It's astonishing how little overlap there is between a given pair of raw files, a given pair of LC-MS experiments for which spectra they contain. So having a nice structured way in which these data are acquired takes some of the randomness out of it. And people have the hope that that makes these, the findings you would produce from these data much more reproducible. Okay, so fixed sampling is a good approach, and frequent sampling is the real winner here. If you're going to quantify something reliably, it's really valuable to have multiple measurements of the precursor intensity and multiple measurements of the fragment ion intensities. And DIA delivers that. So that's a huge win. This allows us to avoid interference. Interference is when two peptides have very similar mass to charge values and very similar retention time values. As a result, you might mistake the intensity of one for the other. This is like the quantitation error we were talking about just a moment ago. Interferences mean that one peptide is interfering with your ability to quantify another. Okay, so does it work? In this case, we have an inter-lab study that was published back in 2019. Each of these different coordinates represents a different lab, and they've been given samples of A and samples of B, and they're both HeLa, lots and lots of human proteins. But spiked in alongside that were 4 proteins that don't appear in HeLa. They spiked in 4 times as much of it in samples B as they did in samples A. So then we asked, is the median ratio that we see in the intensities for these 4 proteins in this HeLa background, are they stably at 4-fold? Because that's how much more they spiked in B than in A. And I think that this is really good evidence that even though these laboratories used really different DIA methods, they were pretty consistently able to get that ratio right at that 1.0 line, which is, because this is log 4, that's exactly a 4-fold estimate. Okay, so in this case, say lab 29, we see that all 4 proteins were observed and quantified, but all of them had a little lower ratio than you would have expected. Now, how bad, what would be unacceptable? In this case, I've opened up this graph to show a fourfold change right along that line, but this 0.5 line, this would mean that the labs claimed it was a twofold change when it was really a fourfold change. And that 1.5 up there, that would be the labs claiming it's an eightfold change rather than a fourfold change. I think we can agree that most of the labs are pretty close to that 1.0 line. They're quite close to the real ratio of these spike proteins. We do have a couple cases where we have high values, but in those cases, those labs claimed that these proteins were up by about sixfold rather than fourfold, which, considering the diversity of methods used by these labs, is a pretty respectable performance, I would say, by a community of people using different instruments differently. So I would say that DIA is actually quite good at nailing down ratios, so long as your protein is, say, in the top 1,000 or so of those that are in a mixture. And that was true for these proteins. The most abundant of them was about rank 600 out of like 3,000 or 4,000 proteins observed and quantified. Okay, brings us to the conclusion slide, which is good. My voice needs a little break. Almost any protein mixture, asterisk, can be identified and quantified by contemporary proteomics technology. That's thrilling, isn't it? You work in worms, you work in soil bacteria, you work in human guts. You can quantify and identify most anything for most any kind of sample. Proteomics has become very, very good at sensitivity. Despite the fact that we're not able to rely on something like PCR, we don't have the ability to photocopy our proteins, but we can still detect them at pretty good sensitivity now in complex samples. Identification relies upon a protein sequence database. If it's not in the database, you cannot identify it. I really love the space of non-model organisms. I have, after presenting this lecture, I'm going to go finish edits on a manuscript that I just got back comments from peer review on about the spotted hyena. Does the spotted hyena have a well-annotated genome? Not so much. There are 4 different genomes that we used in making sense of those data. And it's fun, it's challenging to try to work in a space where you don't have well-documented genomes. So you can do it. But if you don't know that a peptide sequence exists somehow, it's going to be really difficult for you to go about identifying it in database search, because what's in the database is its search space. That defines what it's able to detect. Okay, separating genuine PSMs from false matches is at least half the magic of proteomic identification. We used to think of running the database search as the slow part. These days, so much work has gone into accelerating search engines that most pipelines spend more time doing things like figuring out which PSMs will be accepted and quantifying them than are spent in the database search itself. You'll see an example of that after lunch. Okay. Quantitation can leverage far more than peptide ion chromatograms. Data-independent acquisition quantifies most identifiable peptides. This is shocking to me. I never thought that DIA would be positioned to beat the sensitivity of the old school DDA style experiments. I was wrong. DIA is actually really, really good at producing very sensitive inventories of peptides and quantifying them as well. So I think we've come a long way as a field, and hopefully although we've dragged through a lot of detail, at least the larger outlines of why proteomics is interesting and valuable has come across, not just all the warts and things that come along with the data processing. But I really enjoyed the chance to update this stuff, to think about it from an analytical chemistry point of view, and I hope that it was a useful context for you.
Thank you.
Okay, so did I finish on time? It's 11:52. I'm going to feel pretty good about that. Okay, we're supposed to be back here at 1 o'clock. Does everyone feel they'll have enough time for lunch?
Yep.
I care very strongly about lunch. We're all going to get lunch? Okay, good. So we'll be back here at 1300 for a demo of software.